Hello, welcome. Thank you very much for your interest in our work, our research using AI in biochemistry. I'm Ilan, this is Saima, and this is Madhu. We're doing a triple act today. We're each giving a flavor of our research in biochemistry using AI. And then after that, we'll have a session of questions and discussions, and you're welcome to make comments, to tell us anything. You might be AI experts. We hope you enjoy the next hour. Thank you. We're in the Department of Biochemistry. This is our centenary. And you may have already picked up one of these cards. If not, please have one. It tells you how to engage with our department if you wish. Madhu later will say a few more words explaining it. What is AI? Well, not a day goes by without something about AI on the news, and it's all kind of hyped and frightening. But in essence, it's actually quite simple. It's a machine which essentially mimics, or a machine, a computer, that mimics human intelligence. So it's human-like intelligence. It's not identical to human intelligence, okay? And intelligence is obviously quite a complex concept. And if you're talking about general intelligence, a human dancing and singing and doing music and maths and playing chess, well, we're a long way away from having a machine do any of that, or playing football. But I just want to remind you that machines, i.e. the telephone in your pocket, the supercomputer in your pocket, or any other computer, actually represents everything in binary. That's all it is. It's high voltage and low voltage of a transistor, and everything is represented in binary. Of course, we interact with it at the higher level, which is human readable, human interactable. But everything we're talking about is actually binary. We don't look at it in binary. But if you think of a human brain, which the machine is trying to mimic, it really isn't binary at all. It's a set of neural circuits, and it's actually quite profoundly different from a computer. There's 100 billion neurons, and they're interconnected with all these other neurons, up to 10,000 other neurons. This is a network of profound complexity, and you'll find out in my talk, it's actually continuously organically changing, whereas your phone is just a lump of silicon. That's completely different. So don't get confused. This isn't the same as human intelligence. It's binary mimicking of human intelligence, okay? AI is full of very confusing jargon. So I think later in the talk, someone will mention neural networks. That's an algorithm. And it's a network like neurons are a network, but it's actually more by analogy. It really is not the same as neurons. It's a computational algorithm network. And it's very, very powerful, but it's not the same as neurons. Okay? But the field is just full of all sorts of terminology and jargon. It's quite complex. And actually, us three are not real world experts in AI. We're experts in our own field of biology. And today would be inappropriate anyway to go through this. So some of you may be experts in AI, some not, but these take a lot of work, and they're actually, you need to be programming and doing computer science. So let's just simplify this. Artificial intelligence, or AI, is an umbrella term that includes many computational methods. But because it's only been in the news recently with ChatGPT and so on, you might think it's new. Actually, the term was coined in the 1950s, and the whole field is not new at all. It's been used by scientists for many, many decades. It's just only come to the fore when we've had huge data and amazingly powerful computers. So there's the concept of machine learning, and that's part of AI. It's one branch of AI. And it's fair to say what we're going to tell you about is mostly can be described as machine learning, although some of it's in the news in other forms. But essentially it's quite simple. It's big data, really big data, things like the entire content of Google or all the structures of proteins that we know of. Algorithms that process them in order to find patterns or correlations within the data, either in a supervised way with humans teaching it directly, or without supervision, or with mild supervision, kind of mimicking what the human does. I'll explain one way that happens in a second. And finally, to produce predictions, to find more of the same, or come up with meaning, but it's really important to realize this isn't from nowhere. It requires data, and the data has to be organized in the right way to be mineable. You'll hear from Madhu how really that's a key thing. But it's the organized data, the ability to mine the data is key in all cases. Okay? I'll be talking a little bit about random forest algorithms. I won't really describe that. But it's kind of a series of decision trees that look like a forest of trees. But I'm not going to explain this in detail, but this is just one of many kinds of algorithms like neural networks. Okay, so in the remainder of my talk, I'm going to tell you about how the brain develops from neural stem cells and what molecules are present at the tip of the nerves to regulate synapses during memory and learning, this organic changing of the interconnections. And this is really important for disease as well. So I'm telling you what happens normally, but at the end I'll explain how these processes are correlated in diseases such as Alzheimer's and Parkinson's. Okay, so how do 100 billion neurons and glia, I'll explain what glia are in a second, develop from brain stem cells. So you probably all have heard of stem cells. They're cells which are not differentiated in particular function. They just keep dividing, giving rise to new cells. So like bone marrow is in the news at the moment. Well, in essence, it's quite simple how the brain develops. You have a relatively limited number of neural stem cells in the brain, and these divide to give you, they bud off cells to give you progenitors, and these are kind of replicate, so you get a lot of them, and each progenitor cell then undergoes one division to give you a pair of cells with a specific, unique identity. Because in the brain, the cells have to have the right identity in the right place, and they have to develop at the right time. So the actual regulation of this is really complex, and many labs are working on this. So how are these divisions regulated? Well, I'll just tell you about one study from my lab that's actually already published, where we figured this out in the fruit fly. Now, why the fruit fly? You might think this is so far away from a human, and a fruit fly only has an adult fly only has 100,000 neurons. You might think 100,000, that's nothing, right? Actually, because of this interconnection, these neural networks of neurons and circuits, and the fact that they're all interconnected a lot, this is not binary. This is a huge computational machine. And flies, actually, are really sophisticated organisms, insects in general, and really don't underestimate insects. So the fly has very sophisticated behavior. Even it can fly around this room, avoiding obstacles, finding food, finding mates, doing very sophisticated mating dances. It's really, insects are incredible. Ants, bees, okay? And they only need quite a small brain of about 100,000 cells. But the key thing is about 70% of all fly genes are very, very similar in their molecular function and structure to human genes. So basically, the nervous system evolved a very long time ago, and all complex organisms essentially share the same mechanisms, and the neurons and the glia, the associated cells with the neurons, are very similar. To the extent that a lot of human diseases, say genes associated with Parkinson's, when you mutate them in flies, give you really similar phenotypes, like shaking and discoordination. And the drugs that treat humans can also alleviate those symptoms in the flies. And this is fairly typical and quite remarkable, okay? So this is why we can study the basic processes and the diseases in flies, which is a much simpler thing. But in fact, the fly, the brain of the fly is already fairly developed. The neural stem cells have stopped dividing. So for this reason, we had to go to an earlier stage, which is the larva, which has an even smaller brain, only 10,000 neurons. And in order to watch the neural stem cells dividing. And I'll show you a movie of the division in a second. But the key is, you may think this is a larva, it's a fairly dumb organism. No, it's also really smart. I hesitate to say intelligent, because that's kind of confusing, but this is a smart organism. It can detect gravity, light, it senses smell, it follows food, avoids predators, and it has quite sophisticated behavior. For example, this head-turning behavior. And it needs to do that to survive. But even just doing this crawling behavior requires incredible coordination of all the muscles by the nervous system. So 10,000 neurons is a lot, and it can do all this. And the key is, even that brain, the mechanisms by which it develops and functions are really similar to humans. So what Martin Hailstone described in this paper, he was in my lab, but now he's actually in a startup company doing hyperspectral imaging. He was able to take a larva, which is only a few millimeters long, use scissors which are normally used for dissection of human eyes, okay, in surgery, open up the larva, take out the brain. Now the brain is only less than 0.5 millimeter in diameter, okay, and that's enough to do huge amounts of processing. Open up the larva, put it in an agar dish with the right kind of buffers and so on. Keep it alive for 1 or 2 days. So this is a brain taken out of the body, kept alive. You can't do that in a mouse or human, obviously, or any other kind of complex large organism. And then filmed with high resolution under the microscope. What I mean by film is using fluorescence microscopy, which in essence involves shining a light of one color and that produces light in another color to show you where the molecules are. And that's a technique that's used throughout biology by most labs, really. So when you do that, you get these kind of movies. This is a huge movie. I don't know if you can lower the lights on the screen just here, but this is one section from a huge 3-dimensional movie of an entire brain, okay? And what I'm showing you here is the chromosomes that Madhu will be talking about in red, and cellular cytoskeleton, which technically is called microtubules, but these are fibers that Madhu will show you chromosomes dividing on, but they're throughout all cells and they're used for many things. But here, the key is, are you able to lower the lights? No? You can see these large cells here are stem cells, okay? And I could teach anyone in the audience to identify a stem cell in about 5 minutes, and you'd be able to reliably tell me which is a stem cell, which isn't. And when it divides, it forms this green thing here. And you can see this one's dividing really rapidly, others don't. The key was that you can't really go by hand marking when they divide and when they, what is a stem cell and what isn't, because it's just too much data. it would take you weeks sitting at the computer for one movie. So we needed a method that did this automatically. But we found that no existing image analysis algorithms were able to do this. It might seem strange because humans can just spot it instantly, but this is the power of the human brain. We've evolved to identify patterns, find berries, see faces, just like that. And we're very good at that, and we don't know how we do it. It's subconscious, it's amazing. But computers have to be kind of taught, it's hard. So what Martin did was to build a supervised machine learning image analysis algorithm or software that we named cytosensors to identify what's a stem cell and what's not, and when it divides. And what he basically did is did what I said earlier, take big data, which is a lot of movies to learn from, and then had the user click on a stem cell to teach the machine what a stem cell looks like. And you only have to do about 10, and it learns incredibly reliably. And so this is a non-dividing stem cell. This is a dividing stem cell. That's not a stem cell. And you click on 10 of these, and the machine learns. How does it do that? It basically finds the characteristics of pixels within these images, and that might be brightness, how connected they are with other ones, whether they make a bigger shape that's long, actually literally thousands of characteristics, and then uses these decision trees to decide which characteristics tell you whether it's a dividing stem cell or non-stem cell and so on, and finds the characteristics that distinguish these things. And this is kind of like finding a needle in a haystack, but computers are really good at this. Mindless churning through stuff until you find it. Humans don't do that, yeah? And this algorithm worked fantastically to more than 95% precision. And essentially, it took these movies that it never saw before without training. The training was on a few that we knew the answer for, and basically identifies all the stem cells and then plots their division rate over time. And what we were able to do for the first time is actually quantitate the division rate. And you already see different stem cells in different parts of the brain divide at different rate. This is a slow one, this is a really slow one, this is a really fast one. And this was important because the rate of division decides how many cells you make there, and also consequently what the identity of the cells are. So we then studied mutants where this was aberrant. For example, a mutant where they're all dividing fast, giving you large brain. That kind of is semi-functional, but isn't quite properly functional. And this is the kind of thing that happens in diseases. Okay. So in the next bit, I want to just tell you briefly about another project in the lab involving AI, and that was studying a different question. How are synapses regulated rapidly? So neurons, there's this amazing brain full of 100 billion neurons, basically have these very, very long cytoplasmic extensions. They can be actually a meter long, and they connect up with other neurons via these synapses. But the key to memory and learning is when you make a circuit, when you learn to play tennis and keep doing the same action, some synapses grow to reinforce something, and ones that aren't used shrink. And then new ones are made, some are completely disconnected. But this process is molecular, and it has to occur a long way away from where the DNA is. And this is really one of the key things my lab works on. How is that regulated locally? And the thing that it requires is molecules to be at the periphery to drive this growth or shrinking. And the other thing that we've been really focusing on is glial cells. So glial cells used to be thought of as just a glue that keeps the brain together. But in the last few decades, we've realized they're just as important as neurons. They have long projections, and actually there's very important associations with diseases like Alzheimer's and Parkinson's and other diseases. some diseases, actually devastating diseases, are specifically glial. Okay, so we wanted to know the molecular basis of memory and learning and what goes wrong in disease. What we were able to do was take data that was available in the literature already from mouse and glial and rat glial cells, where people have grown the cells and allowed them to send projections through little pores and then chopped off with a pair of metaphoric scissors the ends, found the molecules in the end compared with the ones in the cell body where the DNA is, and then what we were able to do was take that huge amount of data and process it with a whole load of different kinds of machine learning methods as part of bioinformatics techniques, and basically come up with a predicted set of genes in molecules in Drosophila, in flies, that are at the periphery of glia. And when we did that, these 1,700, we found an incredible statistical association, very, very non-random, compared with randomly found molecules, with all these diseases. And I'm not going to go through all of them, but you'll see Parkinson's, Huntington's, Alzheimer's, ALS, and so on. This was quite remarkable to us. And basically, Maria, who I think is in the audience. Are you in the audience? Yes. Ah, okay, thanks. I couldn't see you. She might rescue me later with any questions which are too challenging. So Maria was able to develop really sophisticated machine learning approaches with random forest. We can, at answer time, we can go through why random forest was chosen to take these 1,700 genes and make predictions of molecules that we missed in this informatics, and also, importantly, additional factors which are required for these molecules to reach the periphery, as opposed to other molecules not to reach the periphery. And I mean, I'm not going to go into the detail, but such a long process requires active transport of molecules with molecular motors. Actually, Madhu will show you a movie of a molecular motor that does this kind of thing. Okay, so the work I described to you involves real teamwork. And most of biology now can't be done by just individuals. It really requires a lot of teamwork. So this is my lab. It involves actually a lot of non-biologists. Maria, whose work I was just describing, who's in the audience, is actually a physics and maths graduate. She came to the lab, learnt biology. There's Danny, an engineer, and so on. There's people from lots of different backgrounds, as well as biologists. The work I described, the data production, which is really the key, was done by these three people. But I really want to give the most credit to Martin and Maria, who built the real AI core of what I talked about. Thank you very much for your interest and listening.
Okay, can you hear me? Yes. Is this on? Okay. So what I'd like to talk to you about now is designing new antibiotics and how I feel that this is going to require artificial intelligence methods. I guess it's going to work. Yeah, so I think, you know, we've already been through one devastating pandemic which was viral in its origins. It turns out that it's very highly likely that, you know, I don't want us to start thinking about the next pandemic, obviously, already, but, you know, the next one that comes along is very highly likely to be bacterial in origin. And the reasons for that is that bacteria are very rapidly becoming resistant to all the, you know, the available arsenal of antibiotics which we have at our disposal. There's very many reasons for that. And actually, I'll go into some of those in a second. And the WHO has, you know, declared AMR, which is a shortened version of antimicrobial resistant, to be one of the biggest threats that faces human healthcare. One of the big problems and the impediments is that pharmaceutical companies don't work on designing antibiotics. And the reason they don't do that is, of course, they can't make money from this. Because as soon as a new antibiotic is discovered, they wouldn't be allowed to use it. They'd have to shelve it and only start using it as a last resort. Because as soon as you start using them, the resistance begins to develop. Right? So it's more profitable to work on in other areas. And so really it's left to charities and the academy really to work on in this area. Luckily for us, a class of compounds called antimicrobial peptides has been identified as a potential candidate or a scaffold which with further development and work could be developed into therapeutics. These are naturally occurring short peptides, they're highly charged, and they're found in most organisms. So we have them already as part of our defense systems. But it turns out that whereas, they won't kill us, they will kill bacteria, and hopefully it's something that we can exploit in the future. Sorry. Okay, so before going into the details, I just wanted to give you, if you'll indulge me for a second, a very quick biology lesson. And I think it's important so we can understand, what I'll discuss in a second. So the reason bacteria are so difficult to kill with antibiotics is that they're protected by a formidable cell envelope. Okay, so compared to us and some of the stuff that Ilan's just talked about, where he's talked about, tens of hundreds of thousands of cells, bacteria, of course, are just a single-celled organism, so one cell, right? No brain, nothing like that, but still we're finding it difficult to thwart them somehow. And the reason largely lies at this cell envelope. So essentially, if we're trying to combat these, we're firing our antibiotics at this single cell. It protects itself by a quite complex surface arrangement. So it's 3 different layers. And you can think of it a little bit like these concentric castles where you have lots of walls which you have to breach before you get into the interior where you want to do your real damage. So these cell envelope antibiotics have to either destroy the cell envelope and kill the cell that way, or they have to get through, breach it, and get on the inside and interfere with some process inside. But one way or the other, they have to negotiate the cell envelope. Okay, so that's what I'm going to focus on. How do we do that with these antimicrobial peptides? Now, unlike Ilan, who's a real biologist, the work I do is computational. So I never go into a wet lab and touch, microbes or I don't even know what they're called, cell lab wet stuff, right? So I do everything on a computer, and essentially what we do is we use computer simulations to make movies that will predict how atoms and then molecules and then bigger organisms, how they work, okay? And behind this movie is a lot of complex mathematics. Right, so it's not just a video game. There's lots of very carefully, well-validated maths that's built on the principles of physics and physical chemistry that goes into doing this. The advantages of using these methods are that actually, they're cheaper, they're greener in most cases. I mean, I hesitate to say greener because we use a lot of electricity and the computers do pump out heat. So they're greener to an extent. And certainly safer than wet lab work. Okay, so that's the reason for using these methods. One of the downsides, however, is that I sort of slightly alluded to, we use an awful lot of computer time just to generate a short movie. So I'll give you an example. We might run a calculation for weeks on the national supercomputer, which is over in Edinburgh. So you'd run it for 2 weeks, and what you'd get out is microseconds of real time. Okay, so it's going to predict what happens in micro. So that's the problem. Because of the numerics, the mathematical number crunching, it's very computationally intensive. Okay, so what can we do with it? So just a reminder, this is what I'm going to discuss. I'm going to show you a movie in a second where we've built a small patch of this cell envelope that surrounds bacteria. Okay, so here we go. Hopefully this will work. So here's one of these movies that comes out from one of our calculations. At the top here, we've got one of the membranes. So this is, if you will, an oily, greasy layer that's on the very outside. And then in between here, we've got a watery, aqueous layer. And in between all these big blobby things you can see are proteins. down here in red is the cell wall, and you can think of this a bit like the skeleton, if you like, that surrounds and gives the cell its shape and some of its rigidity. In green here, I don't know if you just make these out? These little green blobby bits? These are antibiotics. So we've put them in, and we're watching to see how they behave. Where do they go? What do they interact with? And the sort of information we can get out, so if we take 4 different antimicrobial peptides, these are potential antibiotics. And so if we take a look at this one peptide, I can look at a tiny part of what's happening. I can see it beginning to penetrate into this oily layer. And that's great, but it's taken me months of computing time just to get there. And it's predictive and all that, that's all very well and good. But how can I make this faster? Can I speed it up? And so what we're working on now in my group is to see if we can use artificial intelligence. So this is a little, it's called a toddler robot that was developed at MIT. If they can teach a robot how to walk, can I teach a peptide how to move? So we're trying to use the same principles to teach our peptides how to move so we don't have to go through that intensive number crunching. It's literally doing Newtonian mechanics integration at every single step. Instead of doing that, can we take all the thousands of terabytes of data that people have generated in molecular dynamics simulations over the years and use that as a training data set to teach these peptides how to move? And this is one area where we're using AI within my group. Okay, so moving on, I just want to talk you through one other example where we're working towards using machine learning and AI. So I just showed you one example of a tiny part of a peptide and showed that this little peptide inserts into, the membrane, this greasy layer. But what if I want to look at a really, a representative scenario of what really happens? It's not just one peptide. There's, this I showed you here, there's loads of them. I build up my system in complexity. I start adding the proteins. Then I start adding all the small molecules that I know exist inside bacteria, and I put it all together, and I get this horrible complex mixture like this. Now I start running my molecular dynamics simulation, and I try and analyze what I see. And if I want to look at very small parts of it, this is very classical. You'll see this in papers, textbooks, whatever. We can start seeing where the antimicrobial peptide here is interacting with the bacteria, right? Where it's interacting with the native proteins. And I can map these out for very small parts. But because I'm looking at a very big system, and I've got hundreds of these things in there, actually, when I start considering all of them, it begins to look like this. And hopefully you will agree with me that this is now becoming very difficult to interpret by the human eye. It turns out that there's differences between these low salt conditions and these high salt conditions, but unless your eyesight is very much better than mine, I can't see from that. Looking at it, they're not very obvious to me. But there must be a signal in amongst all this noise. Right? And so what we're working towards now is using pattern recognition, so AI methods, the same thing in your phone that allows you to recognize when you take a photograph, picks out features and says, this is a face. Can we use those kind of algorithms to help us pick out the noise in amongst all of this, the signals in amongst all of this data? And all of this now is being fed into a big project we've got with Argonne National Lab in the USA, where they've built up this enormous platform whereby they have a robot that we can do our molecular dynamics simulations, send the details to the robot based on what we've sent it. The robot makes the peptide, it applies it to Petri dishes with the bacteria, comes back the next day, sees how many are alive, how many are dead, feeds that into an artificial intelligence algorithm, which improves on the design of the peptides, gives them back to us, we run the molecular dynamics simulation, and feed the data back to the robot. And we're just about to advertise a postdoc position, so for a worker to come over and work with us and the US on this topic. And so hopefully, the next time we do something like this, we may have some exciting results to report based on this project. Okay, and with that, I'm going to finish by thanking members of my group here. Most of my group is Oxford. I only arrived about 3 years ago, so some of my members, the group members, are still based at Southampton, where I was previously. Whole bunch of collaborators, funding resources, and obviously for us, also computational resources. And thank you for listening.
Right, okay, so can you hear me? Yeah. Brilliant. While Elon sets this up, thank you, Elon. So we've heard about the brain and the bacteria. So in the next 15 minutes or so, we'll go really deep into our own cells. Ooh, that's the last slide. Right. So everybody knows DNA. So DNA has the blueprint for life, right? Oops. Elon. Okay. Has the blueprint for life. So the DNA in our cells is organized into sort of little sausages called chromosomes, okay? So keep that in mind. We'll revisit these sausages in a minute. So the DNA carries the blueprint for life and gets all the credit, but actually the work is done by a group of molecules called proteins. So DNA codes for proteins, and proteins do all the work. So proteins, you can imagine proteins, they're made up of building blocks called amino acids, and it's a sequential chain of these amino acids, okay? And that is entirely coded in our DNAs, okay? So, and different proteins have different sequences, okay? And essentially different chains. These chains then fold up in three-dimensional space and produce a very unique structure. So each protein ends up having a unique structure. As it happens, that is the structure of a hemoglobin protein that carries oxygen in our bloods. Okay, so these proteins that have folded up then tend to accumulate into groups that we call protein complexes, and these groups behave like molecular motors or little robots doing little jobs. So to understand the function of a protein or a protein complex, knowing its structure is absolutely key. So we know the structure of this particular protein complex called kinesin, and knowing that, we were able to really understand its function. So what this kinesin does is essentially carry cargo across the cells, move different things around in different cells, and it does so as shown in this animation, okay? So the key thing I want you to remember here is to understand the function of a protein, you really need to understand its structure, okay? So our work, we are interested in one particular protein complex called cohesin. What is cohesin and why do we study it? So we go back to our sausages. So the whole point of life, the continuity of life, depends on us replicating our DNA, making copies of it, accurate copies of it, and then separating these, splitting up these copies of DNA equally when the cell divides, okay? This process happens about 2 trillion times per day in our body, okay? That's a lot of cell divisions in a day, more or less without mistakes, okay? But it's an incredibly Herculean task. It's okay if you had just one sausage. One sausage becomes 2, you split them up between the cells. Easy, job done. But as you'll know, we have 23 pairs of these, okay? 23 pairs becomes 46, and the 46 now have to be split up between 2 cells. Now, how does the cell know one sausage from another? What if it puts two number 1 sausages in one cell? Disaster, cancer, okay? So we truly need to understand how this works, and it's a massive problem. But I'll give you the solution. The solution is very, very simple. The solution, it turns out, is that when a chromosome divides, instead of it becoming 2 separate things and floating away, it's held together. And because it's held together, these thread-like fibers that Elon talked about in his talk, called microtubules, come from either side of the cell, grab hold of this pair of chromosomes and pull them apart, okay? And in doing so, they enable separation. And as you will see this in this movie, hopefully, so the chromosomes are in red, and these are the human cells that are dividing. And as the cells divide, you'll see that these chromosomes are being pulled apart into sort of 2 equal and orderly piles. Okay? So it's absolutely crucial, therefore, that these chromosomes that are divided are held together. So the question then becomes, what is holding these chromosomes together? How are these chromosomes held together? And here comes our hero, cohesin. This is our protein complex that holds the replicated chromosomes together. It's a protein complex of about 8 proteins, and over almost a decade and a half of work, we believe that 3 of these proteins form like ring-like structure and somehow hold the replicated DNA in there. But the thing is, we don't have the structure of the entire complex. And if you remember, I told you already that in order to understand the function of a protein, you have to really understand its structure. So why do we care? Because as I've already told you, because if the chromosomes aren't held together, there is no way life can be sustained. And when things go wrong, they go catastrophically wrong. And cohesin is one of the highly mutated genes in all of cancer. So, for example, 50% of all lung cancers and melanomas have defective cohesin molecules. So we truly need to understand how this works. And in order to do so, we need to get a handle on the structure of the complex. How does the ring work? How does the DNA get in? How does it end up holding the divided chromosomes together? So how do we then get the structure? So theoretically, it is possible to predict the structure based on the sequence, based on these beads on the chain. But as Simon alluded, it is an astronomical number of possibilities that these things can fold up to. If you imagine I give you a chain of about 500 beads, you can literally make millions of different ways in which you can fold them. Yet in a cell, it's only one that is stable. Every protein has one unique structure. So how do we pick that out from all these millions of different possibilities? So therefore, traditionally what we've been doing till now is to get the structure of the protein empirically. So what this means is that we can induce these proteins to form these crystals or little lattices, and we can shine X-rays onto these crystals. And then by looking at the way the X-ray is scattered from these protein crystals, we can get an idea of the structure of the protein. Okay? So this is what's been happening thus far. But it's an incredibly time-consuming process. And to get at a protein structure, you have to cross a moat filled with the PhD students' tears. So far, it's been one structure, takes about 4 to 5 years, essentially one PhD. Okay? And the other problem is that not all proteins are amenable to crystallization. You can't make them into lattices, large proteins, floppy proteins, the ones that are really flexible, and proteins that Simon talked about, membrane proteins that are greasy a little bit. They don't tend to become crystals. So it's a huge problem. But we do have about 200,000 of these protein structures that we've solved over the past 50 years. And with some remarkable foresight, what we've been doing is to deposit all these protein structures into a database that we call Protein Data Bank, and that's freely available. Okay? So here enters the AI. About 4 or 5 years ago, a company called DeepMind, which is now called Google DeepMind, thought it would take up this challenge of predicting protein structures. Okay? So they came up with an AI tool that they call AlphaFold. It is essentially a neural network. Elon explained what neural networks are. And this neural network has been trained on this database of PhD students' sad story called the Protein Data Bank. And it has learned how to predict structures. So how does it work? This is a complicated flowchart of how AlphaFold works. But to put it in simple terms, it takes this sequence of beads on a string that you put in as input, and the first thing it does is it compares to all known proteins and finds similarities. And this information is then passed on to another neural network that tries to predict angles and distances between two beads, if you will, on the string. And by compiling all of this, the program is able to give you a prediction of a three-dimensional structure of the protein. Okay? So what used to take about 4 or 5 years can be done within minutes by this program. Okay? But is it any good? So as you will see, these are two predictions superimposed to actual structures of these proteins that were determined empirically. So what you see in green is the actual structure, and blue is the prediction by AlphaFold. And I think you'll see that they're pretty much bang on. So it's truly remarkable how accurate this AI software is in predicting. And over Christmas, about 2 years ago, they went and predicted the structure of all the proteins in our body. So, and in about a year's time, they've predicted about 200 million protein structures and made everything completely freely available. So like a Google search, you can go onto this website, type the name of your favorite protein, and get the structure within seconds. Okay? So this has completely revolutionized biomedical and biochemical research, because it gives you the structure. You can understand function. You can start to understand function. And it has opened up completely new doors. You can screen for drugs. You can do vaccine development, everything in the computers, even before you enter the lab. So if you're interested in that, we can talk about it during question time. So we go back to our problem, which is understanding how chromosomes are separated or held together. Go back to the problem of cohesion. So I told you that we don't have a structure of the entire complex. So what did AlphaFold do? It went and showed us that we were indeed right, that the cohesion complex does look like a ring, and it is conceivable that the DNAs get trapped inside. And we didn't have, I told you it's a complex of 8 proteins, and we didn't have structures of most of those. It gave us structures of all the proteins, and it told us where each of these proteins are assembling onto the ring. So this has completely changed our future perspective. And this is crucial because one of these proteins we know is the most highly mutated gene in all cancer. So this is where we are now, and this AlphaFold has completely given us a whole bunch of new opportunities to try to understand how this amazing protein complex works. So I'll end here and thank all of the people in my lab. So if you want to find out more about our work, please come into the department. We're always happy and welcoming. And as Elon mentioned in the beginning, this is the centenary year of our department. We are celebrating 100 years of amazing research. And as I said, if you might have picked up a card while you come in. If you haven't, please get one on your way out. And in the back, there's information about how you can get in touch with us if you want to know more, if you want to engage with our research, come visit us. And in December, we are producing a booklet that is free for people who show interest, who can come in and get it for free. And this booklet is going to detail amazing stories about scientific research that's happened in the department over the past 100 years. Thank you.